Hello students and welcome to another video. I'm your teacher, Mr. Flynn, and today we're going to be showing you how to do the microscope lab. Now, there isn't going to be questions associated with this. It's just you showing me competency when using a microscope. I have a previous video on this and it goes through the anatomy and the different parts of a microscope and how to use it. But today I'm just going to show you how to set up a slide and what is expected for you to show competency to show me how to use a microscope, okay? Because we're gonna use this several times through the year, but I need to have you show me that you can do it. All right, now how we're gonna do that is each station is gonna have a glass slide that looks like this, if you can see it angled in the light there. Now, there's a frosted end. Now that's where you hold the slide so you don't keep your fingerprints and oils on the slide, but we're just gonna be doing some simple things, so it's not a huge difference, okay? So when we're holding slides, just hold it like that. We also have a cover slip that is made out of plastic, and these are going to go together, okay? The glass slide is where all of your specimens are, and then the cover slip goes on top of your specimen, okay? Now the specimen we're gonna be looking at is a newspaper clipping or a magazine clipping. So you're gonna cut out one word from the magazine, okay? And you're gonna place it onto your slide like this. Now, when you put the cover slip on, a lot of you will want to set it straight down. Okay, don't do that. I want you to pretend there's a hinge. So hit it on the edge and then let it fall this way. All right, the reason we do that is to prevent air bubbles from forming. But because this is a dry mount, it doesn't really matter. But afterwards, you're gonna do it with water, and so that does matter to do it a certain way. So just out of habit, just pinch the one end down and let it fall onto it, okay? Okay, oh, now because it's dry, all right, things fall off very easily. All right, now you'll realize quickly why wet mounting is better. Because of that cohesive and adhesive force of water, it kind of keeps it all together. So what you'll do is you'll slide it underneath, okay? But make sure it's under the scanning power first. That way you have room, okay? All right, so put it like this, all right, and get it lined up so that's right underneath your lens. And you have stage knobs where you can adjust the movement on top of the stage, okay? These larger and smaller knobs are for moving the stage up and down so that you can focus your lenses, okay? All right, so now it's all set up. You get it centered. All right, you find a nice thing to look at. I want you to work your way all the way up from the scanning to the low to the high power. And then come show it to me, and then I will check you off. All right, and then I want you to do it again, but with a wet mount. Now, if we look under here, okay, you'll see that the highest power lens, I'm gonna exaggerate it for you. If I drop my stage all the way down, okay, and there's a gap between the stage and this high-powered lens, you're doing it wrong. You've got to start over. You got to start back at the scanning. Your blue lens should just barely be touching the stage and your slide that you've set up, okay? So pay attention to that. So if I come walk over and you have a big gap between your high power and your stage, I know you're doing it wrong just by looking at it, okay? So make sure that it's right up against it, okay? All right, so to do a wet mount, here's how we do that. I'll show you this, and then the wet mount's gonna be the same thing where you're gonna get it up to a high power, and then I'll check you off. And you will see a difference between a dry ink versus wet ink because the water's gonna absorb into the magazine clipping, and what you'll see is that black ink will actually start to break apart and you'll see cracks in it. So I want you to kind of see before and after, and you'll see that wet mounting is actually preferred. So I'm just gonna take my clipping and put some water on here, just a drop or two, and then take my cover and I'm just gonna lay it on hinge-like, okay? I'm just gonna set that on there to prevent all those air bubbles from forming, and then I'm gonna set it on my slide, okay, with it on the scanning. I have it low, so just make sure that's on the scanning so it gives you room. Now, don't be adding water while it's on the stage. I want it to be on the table and then you can put it on. Don't do experiments on the stage, all right? Okay, then make sure that when you plug it in and unplug it, that you're holding it by the top of it. Don't be yanking on my cord. And sometimes the outlets, they need to be reset. You'll see two buttons. They're often like the hair dryers that you guys use at home. They have a little breaker on them. So if you're using them and it's not turning on, hit those buttons and hit the test and then reset it so that the power can flow into your microscope, okay? Thanks again for watching. Till next time. Oh, actually, one more thing. When you are done, I will have extra slides around the room. And then I want you to look at muscle cells, red blood cells, and spend some time going through and looking at other specimens rather than just newspaper clippings, okay? And then getting better at just using the microscope in general, okay? Thanks again for watching. Until next time.